the treatment that we have, PBGM01, is a gene therapy that uses what we call the adeno-associated viral vector, or AAV, which has been, we're in our partnership with the Penn gene therapy program. Jim Wilson, who leads that program, is really sort of a pioneer in the AAV gene therapy world. And the specific form of serotype of AAV that we're using has tropism for neurons. And so what we do is we actually, in contrast to some of the other gene therapies with AAV that are approved, which are done systemically, we actually administer the drug into the CSF directly at the base of the brain, into the cisterna magna. And what we have found in our work with Jim Wilson and his team at Penn is that we get much better distribution of the viral vector throughout the brain at much lower doses than we would have to use if we did systemic administration. And so those viral vectors are transfect cells, which leads to the expression in the neurons of the brain. It's then, and actually it's only a small percentage of the neurons that are actually getting transfected, but the protein is secreted, but then can be something we call cross correction, is that other cells that can take the enzyme up and utilize the enzyme. So you get, even though you're only transfecting cells at a small percentage of the cells, you actually can get very broad distribution of the transgene specific transgene, in this case, beta-galactosidase, throughout the brain of the patients. And I mean, so we have, this was demonstrated very nicely in non-human primates, and now we have to see if that translates into effects in humans, which is where we are. We're running our very first, for what we call first in human trial with our vector.